This video will cover the action potential, resting membrane potential, depolarization, repolarization, neuromuscular transmission, and excitation contraction coupling, including the mechanism of muscle contraction and the sliding filament theory. An action potential is a rapid, brief, and intense change in the membrane potential of a cell, from a negative resting potential to a positive peak, and back. This change is triggered by the opening and closing of ion channels and is the basic mechanism for nerve and muscle cell communication. The resting membrane potential is the electrical potential difference across the plasma membrane of a cell when it is not actively communicating. It is typically around -70 millivolts for nerve cells and -90 millivolts for muscle cells, maintained by ion concentration gradients and ion channels and pumps. All cell membranes have a sodium-potassium ATPase pump that transports 3 sodium ions outside for every 2 potassium ions inside. This is an electrogenic pump, resulting in a net deficit of positive ions inside and causing a negative potential. Potassium leak channels are channel proteins in the nerve membrane through which potassium ions can leak, even at rest. These channels are about 100 times more permeable to potassium than to sodium. Depolarization is a change in membrane potential where it becomes more positive, often due to the influx of positively charged ions like sodium. In nerve cells, this initiates an action potential, while in muscle cells, it triggers contraction. Repolarization follows depolarization, making the membrane potential more negative again to return to the resting state. Sodium channels close, potassium channels open, and positive potassium ions leave the cell. This causes muscle relaxation in muscle cells. Voltage-gated sodium and potassium channels are crucial for depolarization and repolarization. The voltage-gated sodium channel is the necessary factor for both, and the potassium channel increases the speed of repolarization. The voltage-gated sodium channel has an activation and an inactivation gate. At the resting membrane potential of -70 millivolts, the activation gate is closed. When the potential rises towards -55 millivolts, the activation gate suddenly opens, allowing sodium ions to pour inward. The inactivation gate closes shortly after the activation gate opens. This stops the influx of sodium ions, and the membrane potential begins to return toward the resting state. This gate will not reopen until the membrane potential is near the original resting level. In the resting state, the voltage-gated potassium channel is closed. When the membrane potential rises, this voltage change causes the channel to open. They open as sodium channels begin to close, speeding up the repolarization process. Neuromuscular transmission allows the central nervous system to control muscle movement. Nerve impulses release acetylcholine, a neurotransmitter, into the junction between the nerve cell and the muscle cell. Excitation contraction coupling converts an electrical signal into a mechanical muscle response in skeletal and cardiac muscle cells. Ion movement generates the signal, triggering calcium release from the sarcoplasmic reticulum. Calcium ions activate contractile proteins, causing muscle contraction. Muscle contraction begins when an action potential travels along a motor nerve, releasing acetylcholine. This neurotransmitter opens cation channels, allowing sodium ions to diffuse inward, causing depolarization that initiates an action potential in the muscle fiber. The action potential travels along the muscle fiber, causing the sarcoplasmic reticulum to release large quantities of calcium ions. These ions initiate attractive forces between actin and myosin filaments, causing them to slide and contract the muscle. Calcium ions are then pumped back into the sarcoplasmic reticulum and contraction ceases. The sliding filament theory explains muscle contraction at the molecular level. It states that actin and myosin filaments slide past each other to shorten the muscle fiber. Acetylcholine release triggers myosin heads to bind to actin, forming a cross bridge. Myosin heads use ATP energy to pivot and pull the actin filament toward the sarcomere center, causing muscle contraction. This process is also known as the sliding filament theory. Muscle contraction is achieved by the sliding of actin and myosin filaments. In skeletal muscles, an action potential releases acetylcholine, causing depolarization and calcium ion release. Calcium binds to troponin, and actin and myosin interact, resulting in muscle fiber shortening.